Assalamu alaikum, second year. So previously we learned about the labyrinth, its structure, the compartments of cochlea and the fluids inside them, and how the endocochlear potential is generated. And in the lectures before that, we have gone through the overall events in the hearing. Uh, you all must know that the mechanism of hearing, it can be broadly classified into three steps. The first one is the mechanical conduction of the sound. Then the next step is the transduction of this mechanical energy into the electrical impulses. And finally, these electrical impulses, they are conducted to the brain. So what actually happens that when sound waves, they arrive at the tympanic membrane, there is a movement of the tympanic membrane. Now this movement, it causes the displacement of the auditory ossicles, as I already told you. And the final bone in this ossicular chain is stapes. The movement of the stapes at the oval window, that establishes pressure waves in the perilymph. The perilymph, which is present in the vestibular duct, which I told you in the previous lecture. Now, this pressure wave in the perilymph, it distorts the basilar membrane on its way to the round window of the tympanic duct. So it generates the vibrations in the basilar membrane. And these vibrations, uh, they cause the vibration of the hair cells against the tectorial membrane, as a result of which there is activation of the hair cells. Now, this excitation, it generates nerve impulses. The information in the nerve impulses about the region and the intensity of the stimulation, that is related to your central nervous system, and that is relayed by the cochlear branch of eighth cranial nerve. So that is the overall mechanism of hearing. Right. So, what are the learning objectives of today's lecture? Today, I will tell you that how the transmission of the sound waves that takes place in cochlea. Uh, the most important thing that you will learn in this lecture is that how a traveling wave is initiated in the fluid of the inner ear. And you should also learn that what is the resonance point, and then you will see in this lecture, you will see the vibration pattern of the basilar membrane, and I would like you to focus on how the waves of different frequencies, they travel differently along the basilar membrane. Then we will look at the pathways that this wave takes, and you should also focus on the transduction mechanism that takes place in the cochlea. So let's start with our today's lecture, which is about the functioning of the inner ear. Earlier, we did our the physiologic anatomy of the inner ear. This lecture is about the functioning of the inner ear. Now, this function it is divided into two parts. The first part is the sound transmission, and the other is the transduction. So, first we will start with the sound transmission. The transmission of the sound waves in the cochlea that takes place in the form of a traveling wave. Uh, you all know that cochlea that is bounded, it is limited on all the sides by the bony walls. So when the foot plate of the stapes, the face plate of the stapes that moves inward against the oval window, then the round window it must bulge outward. You see that when the oval window it moves inward to get rid of this pressure energy, this round window it must bulge outward. Now the question is that how this traveling wave is initiated. The initial effect of sound wave entering at the oval window, that is to cause the basilar membrane at the base to bend in the direction of the round window, as a result of which an elastic tension is built up in the basilar fibers because they bend toward the round window. Now, this elastic tension it initiates a fluid wave that travels along the basilar membrane towards the helicotrema. Now, this movement of the wave, which is along the basilar membrane, you see that it is comparable to the movement of the pressure wave along the arterial walls. So, what happens? That once it bends towards the round window, there is elastic tension. And because of this elastic tension, a fluid wave is generated which travels along the basilar membrane towards the helicotrema. Now, what is the resonance point? It is the part of the basilar membrane which is activated by the traveling wave to a maximum extent. At this point, you see, this is the resonance point for say one frequency. Now, this is the resonance point for another frequency, and this is the resonance point for still another frequency. So at this resonance point, the wave it stops here and it does not travel any further. Now you see the distance between the stapes and the resonance point that is inversely proportional to the frequency of the sound waves. The distance between the stapes and the resonance point that is inversely proportional to the frequency of the sound. So, if the frequency is higher, it means the distance will be shorter. And if the frequency is lower, the distance will be bigger. Right. So now we will have a look at the pattern of the vibration of the basilar membrane. Each wave that is relatively weak at the outset, but it becomes strong when it reaches the portion of the basilar membrane that has a natural resonant frequency equal to the respective sound frequency. So, actually, what happens that the wave at the starting it is weaker, but later it becomes stronger, and it is strongest at the resonant point of that particular frequency where the basilar membrane it it has the equal resonant frequency. uh, so we can say that the high frequency wave that travels only a short distance, the medium frequency wave that will travel about you can say halfway, and the low frequency wave that will travel the entire distance. Right. The speed of the traveling wave it also has some variation. The sound wave it travels fast along the initial portion of the basilar membrane, but it becomes progressively slower as it goes farther into the cochlea. Why is that so? It is because of the high coefficient of the elasticity of basilar fibers, which is near the oval window. And progressively, this coefficient of elasticity it decreases further along the membrane. Now, this difference is not without the advantage. You see the rapid initial transmission of the wave it allows the high frequency sounds to travel far enough into the cochlea to spread out and separate from one another on the basilar membrane. If there wasn't any difference in the speed and there is no rapid initial transmission, without it all the high frequency waves they would be bunched together within few millimeters, and then their frequencies they couldn't be discriminated. Right. So next we move on to the pathways which our traveling wave takes. uh, you already know that there is fluid movement within the perilymph, which is set up by the vibration of the oval window. Now, this wave of the fluid movement, it follows two pathways, as you can see in the figure. One pathway is longer than the other, Now, the first pathway, which is labeled here, this is the first longer pathway that is through the scala vestibuli, around the helicotrema, and through the scala tympani, and it causes the round window to vibrate. Now, this longer pathway, this is just to dissipate the sound energy. This is just to dissipate the pressure which was built up here at the oval window, right? The second pathway, which is starting from here and then it moves like this, it's a this second pathway, it's a shortcut, and that is from the scala vestibuli through the basilar membrane and then to the scala tympani. Now this pathway, this shorter pathway, it triggers activation of the receptors for sound. And how does it do it? By bending the hairs or the stereocilia of the hair cells on the organ of Corti, which is present on top of the vibrating basilar membrane. Because this basilar membrane that is displaced in relation to the overlying tictorial membrane. So this is how this shortcut it causes the generation of the nerve impulses from the hair cells. Right. Oh, I might strike your interest back into the lecture by introducing you to George van Bekassi. He proposed the traveling wave theory and he won Nobel Prize for this in 1961. Right. So now we move on to the second part of the functioning of inner ear, which was transduction. Certain electrical events they take place in the cochlea, such as the generation of the receptor potential in the cochlea. What is this receptor potential? It is the you can say it is the mild depolarization which is developed in the hair cells of cochlea. Now, this is a microphonic potential. It is the cochlear microphonic potential. Now, this cochlear microphonic potential that is graded potential. And this graded potential is called the receptor potential of the hair cells. Now, this receptor potential, it is non-propagative, but it can cause the generation, in fact, it causes the generation of action potential in the auditory nerve fibers. So we will look into the detail of the generation of this receptor potential. As I already told you that there are lateral crosslinks, or you can call them tip links, which attach the stereocilia to one another within and between the rows. You see that these links, these tip links, they are attached to mechanically gated channels also, which are present on the stereocilia. And these channels, they open when the tip links, they stretch. And when do they stretch? On bending of the stereocilia towards the kinocilium, towards the tallest stereocilium. So when the other stereocilia they bend toward the tallest stereocilium, these mechanically gated channels, they open. So the movement of the stereocilia towards the kinocilium, it causes depolarization of about you can say minus 50 millivolts. The inner inside of the membrane, it reaches from minus 60 to minus 50 millivolt. So they cause the depolarization in the hair cells by the opening of these mechanically gated channels. And these mechanically gated channels, these are the potassium channels. Right. So upon the depolarization, the hair cells, see here, that the hair cells, they release a neurotransmitter, which could be glutamate also. And this release of the neurotransmitter, that generates the action potential. Now the movement in the opposite direction, that will cause hyperpolarization, and this hyperpolarization, that stops the generation of the action potential in the auditory nerve fibers. All right. So let's have a look at these figures, which are showing the you know the depolarization and the hyperpolarization of the receptor hair cell. And you can look at the stepwise mechanism for the generation of the cochlear potential. You see, in the first step, the if we are talking about the depolarization, in the first step, the tip links, they stretch and they open the channels when the stereocilia, they bend toward the tallest member. So when the channels open, the potassium ion in the endolymph, it it enters. There is influx of the potassium ion, and the hair cell, it depolarizes. Now the depolarization of the hair cell, that opens the voltage gated calcium channels here. Now this calcium ion entry, it causes a greater release of the neurotransmitter because they push the neurotransmitter vesicles towards the prejunctional membrane. They fuse with this prejunctional membrane and then they release the neurotransmitter in the synaptic cleft. So this greater release, the more neurotransmitter, it leads to a higher rate of the action potential. So it means that in resting condition also there is a release of the neurotransmitter and there is generation of the action potentials, but there is low rate of the action potentials. On depolarization, the rate of the action potential, that is increased. And on the other hand, when the tip links, they, in case of hyperpolarization, the tip links, they just slack and they close the channels when the stereocilia, they bend away from the tallest member. So no potassium enters and the hair cell, it hyperpolarizes. No calcium enters and no neurotransmitter release. As a result, there is no action potential. So this is how the action potential is generated. The nerve impulses, they are generated from the hair cells. Here you can go through the entire process of the overall mechanism for the generation of the nerve signals and how they are propagated to the CNS. You see that the sound waves, they cause the vibration of the tympanic membrane. Then there is conduction of this vibration through the middle ear bones to the oval window. As a result, there is vibration of the oval window, and this vibration, it causes the fluid movement within the cochlea. Now this movement, it causes the vibration of the round window and also of the basilar membrane. The movement of the basilar membrane, that causes the displacement of the hair or the stereocilia, that is in relation to the overlying tectorial membrane. So this movement, this results in the bending of hair or stereocilia of the inner hair cells. Now this bending, this causes the changes in the receptor potential, and finally a change in the rate of the action potentials in the auditory nerve fibers. So this is the whole mechanism of the generation of the action potential in the hair cells. So I hope you are clear about this thing. In the next part, we will start from the nerve impulses that once they are generated, how they reach to the CNS, and what do they show